Hi everyone, I'm Dr. Mark Hackett, a lecturer at Curtin University, and welcome to one of our labs here in chemistry at the Curtin Bentley campus. In today's experiment, we are going to be measuring proteins, and specifically we're going to aim to determine how much protein is in an unknown sample. Proteins are essential for biology. They're really important to all of our cells in our bodies, as well as cells in plants and animals. For example, there's special types of proteins called enzymes. These enable specific chemical reactions to take place in our body. There's other proteins that are used to actually shuttle around molecules involved in energy production, as well as molecules such as oxygen. There's also proteins that are used as scaffolds. These provide the framework to actually build our cells and tissues. So it's really important for scientists to be able to actually measure how much protein is contained within a sample. So a protein is made up of a special covalent bond that exists between the amine functional group and the carboxylic acid functional group in an amino acid. So when the amine functional group reacts with the carboxylic acid functional group, we eliminate a molecule of water and form an amide or a peptide bond. And we can use the chemical properties of that peptide bond to actually determine if a protein is present and how much of the protein is there. The reaction we're going to use today is the buret reaction. And this uses copper ions and a color change to determine if a protein is there. So when we have copper ions in solution, they appear blue. So there's a chemical complex formed here between the water and the copper. If we add protein into that mixture now, the lone pair of electrons on the nitrogen atoms in the peptide bond are able to displace the oxygen atoms. As that occurs, there's a color change, and we go from a blue color to a more purple color. So we're able to use that to identify that there is protein present in the sample, and the intensity of the color of that purple is proportional to how much protein is there. So the more protein we have in our sample, the more displacement of the water oxygens there are, and the more nitrogen will bind to the copper, giving a darker purple. Okay, so let's get started. So to begin with, we are going to prepare our protein standard. So we're aiming to make a 10 milligram per mil standard solution of protein, and we're going to use gelatin as our protein standard today. So we have 1 gram of gelatin that we've accurately weighed out on the balance behind me here, and we're now going to dissolve that in 100 mils of water. So we're now going to stir our gelatin protein solution until the protein has all dissolved. We're now going to prepare our unknown samples. So here we have some egg white dissolved in water. We're now going to prepare our buret solution. So this is the solution that's going to start as a pale blue color, and then as we add protein to it, is going to turn into a lilac or a purple color. And the intensity of that color is going to tell us how much protein is there. So the more protein, the darker the purple or lilac color. To make our buret solution, we're going to start with 0.75 grams of copper sulfate into a 250 ml beaker. We're then going to add 3 grams of Rochelle salt, so potassium sodium tartrate, and we're then going to start by dissolving this in some deionized water. So once we have dissolved our salts in this beaker, we're going to add sodium hydroxide. So we're using relatively dilute sodium hydroxide, 2.5 molar. However, it is still caustic, so you may wish to put on safety gloves for this start part of the experiment. So we can now work with our sodium hydroxide and add that to our buret reagents wearing our safety gloves. So here's our final product of the buret solution. So this has our copper sulfate, our Rochelle salt, and our sodium hydroxide added. And we're now going to make it up accurately to the 250 ml mark using deionized water. So I'm going to come down to eye level and bring the bottom of the meniscus up to our 250 ml mark. And we can now shake and stopper that. We're now going to add our buret mixture into our calibration samples. So we've prepared 11 calibration samples here containing a range of protein concentrations. So in calibration sample 1, we just have deionized water, so no protein, all the way up to 10 milligrams per mil in calibration standard 11. So we're going to add to this now 4 mils of our buret solution. So take our pipette. So this is set to dispense 1 mil at a time. So we'll dispense 4 aliquots. I'm going to pour out a sample of the buret mixture. So I'm going to dispense now into our first calibration. So this sample doesn't contain any protein. This is our blank, so we shouldn't see any color change. We've added our 4 mils of our buret solution. Give it a gentle swirl and a tap. Now we should wait approximately 10 minutes for the reaction to complete, but hopefully you can see that stayed this light blue colour. So while that waits for 10 minutes for the reaction to complete, we'll do the rest of our calibration standards. So now we've added our buret reagent into each of our calibration standards. But here's our first standard. So this doesn't contain any protein, and we can see that that has maintained the light blue colour of copper ions in water. In our next sample, we have 1 milligram per mil of protein there in this standard. So we can see the light lilac purple colour appearing. At 10 milligrams per mil protein, the sample on the right, we have the darkest purple of all. So in this case here, we're using our eyes and our eyesight as a spectrometer. And the role of that instrument is to identify and differentiate between different wavelengths of light and to determine how much light there is. Rather than using a spectrometer as an instrument in this lab, we're actually using our eyesight to do that. So to identify the colour purple and visually measure how intense it is. So now that we have our nice calibration along the front, we're going to add the buret reagent. So I'm adding in 1 mil of our egg white, dispense that pipette, and now I'm going to add 4 mils of our buret reagent. So what we expect to see is a purple colour forming. As we all know, there should be lots of protein in eggs. So this is really cool. We can see a nice purple colour that's come from the protein in the egg white reacting with the buret reagent. So we're going to let this stabilise for 10 minutes. Now that we have waited 10 minutes and the colour has stabilised, we see this nice intense purple colour from the reaction between the egg white proteins and the buret reagent. So we'll compare. So this was 7 milligrams per mil protein on the left, and it's almost an identical colour to our egg white sample. So we can conclude then that there's roughly 7 milligrams per mil protein in our egg white sample. We hope you've enjoyed learning about how to measure protein today and found it knowledgeable. If you would like to find out more about studying chemistry or a science degree at Curtin, please head to study.curtin.edu.au.